Hi, I'm Smayan and today we'll learn about genetic engineering. So, have you ever wondered that how these superheroes do all these things? Like Flash does all these running moves and all these. Well, now you can get this same thing in your body through a simple process called genetic engineering. So let's get into the basics: the history, the current modifications, and the future uses. So first, let's go with the history. So the history of genetic engineering is that humans have been genetically modifying organisms for thousands of years. For example, let's say the bananas. Bananas used to be hard and had a green outer shell with thick seeds within them. And through genetic engineering, we have used them and turned them into these delicious yellow versions. And you could say the same with dogs. Dogs used to look a lot like wolves, but through genetic engineering, they basically become what we call now puppies. Very cute. Anyway, let's get into it. Now, genetic engineering is basically the modification of genes in your body. Basically, their DNA is deoxyribonucleic acid, the code of life. So, it is a very complex molecule that controls your development, your reproduction in your entire body. Think of it like the blueprints of your body. Change the blueprints and you change the being carrying it. So genetic engineering was first introduced. Well, mainly it was found, but okay, let's say introduced in the 1950s. So as soon as it was found in the 1960s, people started tinkering with it. They used radiation on plants, and basically they just turned them into genetically modified organisms or GMOs, but they deserve a video on their own. So basically they started it through radiation, and they turned plants into these genetically modified organisms. Basically the idea was that if they used it, they could get a useful plant variation out of complete chance, and sometimes that actually worked. But until recently, this process has taken a lot of money, time, and development. Until recently now, there is a revolution called CRISPR, clustered regularly interspaced short palindromic repeats, or CRISPR for short, which is basically a new way of how we can genetically modify organisms. So this was first found by Jennifer Doudna, and so this is how it worked. So bacterium and viruses have been fighting since the dawn of life. Bacteriophages, or phages for short, hunt down the bacterium and basically turn them into a factory for a virus explosion. And in the seas, viruses called bacteriophages destroy about 40% of all bacteria in the sea. So basically, these phages usually disarm the bacterium. The weak defenses of the immune system usually cannot do anything about it. But in some rare cases, that actually does happen. And the bacterium actually defeats the virus. Until then, only the real fun starts. Then, the bacterium can arm a secret weapon. So basically, the bacterium saves a secret save file of the virus's DNA in its own DNA archive. If the virus ever does attack again, it will be ready for it. It'll scan for the same splice of DNA from the sample, and if it does find, it will arm a secret weapon called Cas9. Now, this weapon of the bacterium can disable the virus's DNA and the virus itself, because the viruses actually insert their own genetic sperm. Basically, the Cas9 system can latch onto it and deactivate it by repairing it with a new RNA strand, by quickly making a copy of the DNA strand called RNA or ribonucleic acid. Then it comes in and basically just deconstructs and basically vaporizes the entire DNA. And that's how the bacterium has gained complete immunity of the virus. Now, in the real fun story, in 1940, in the 1950s, where people, not 1950s actually, I'm just recently, when they found out that the CRISPR system was actually you could actually program it, meaning you could manually control it. It was so simple that if you just gave it a strand of DNA and put it into a organism, it would do everything for you. Basically like a precise surgeon. If the old ideas of genetic modification are correct, then genetic modification can be said as a map. In that case, then CRISPR can be used as a GPS for that DNA system. So basically, what we can do is use organisms from different animals or other organisms. Say, for example, you want to run as fast as a cheetah. Well, a special gene in it, if you take out that gene and use it in your own body, you could basically get the genes of the cheetah, which will allow you to get the really powerful legs of the cheetah. And then you can run really fast, as fast as the cheetah, which is about 50 kilometers per hour. So yeah, now we can use this for some of our greatest problems, like malaria. We can suppress the genes of mosquitoes by adding a antibody gene into its own archive, which basically immunizes the mosquito from plasmodium, which is basically the virus for malaria. Now, what we can do is use it to control our greatest threat ever: death from aging. Now, there are some animals that can never die, like the immortal jellyfish, the lobster, and the hydra. Now, what we can do is use these genes from these organisms and implement it into our own chromosomes, meaning the telomeres of our chromosome. You see, in our cells, we have telomeres on our chromosomes. You see, each time a cell duplicates, these telomeres get cut off a bit, basically so that no damage comes to the chromosome. But so basically, the telomeres can regenerate easily. But over time, this regeneration process wears down, and then soon the telomeres wear down. Then you start getting a whole heck of symptoms associated with aging, because now your telomeres are gone and the chromosomes are exposed to harm. So basically, other organisms have special telomeres that allow them to basically never get their telomeres worn off. Now we can use the genes from these organisms and implement it into our own body, and therefore we will gain immunity to aging, which could be really useful to humankind. That's all from Smayan Sahu, and I will bid you all farewell. Thank you.